Hi, my name is Muhammad Zawawi and this is my outstanding performance for the final year report, which I'm going to talk about implications of sterilization techniques in plant tissue culture of Herbaceous plant. Before we go through with the main idea, let's go with the definitions. Have you ever washed your vegetables before cooking or sanitized your hands after get out of a supermarket during this pandemic of COVID-19? Well, that is called as disinfections, which is a process eliminations of pathogenic microorganisms, as it is not effective in case of vegetative spores. Sterilization is known as statistically complete removal of germs, including the most resistant bacteria and spores, while plant tissue culture is culture that produce plant clones known as micropropagations of in vitro cells, tissues, organs, or plants under sterile conditions. So in this video, we are going to analyze the result of the effect of sterilization techniques toward the plant tissue culture in terms of its physical and chemical property, and compare the sterilization procedure in the applications of plant tissue culture of Herbaceous plant. 6 germinations, embryo culture, meristem tissue culture, enter culture, and callus culture is sort of Herbaceous plant tissue culture in which the explants of a certain plant species is extracted and sterilized before being added to the medium. Now let's go with the type of sterilizations and its effectiveness. The first one is plant tissue culture instruments sterilization, which includes autoclaving, dry sterilizations using ultrasonic cleaner, flame sterilizations, and infrared sterilizer. Those sterilization techniques or method use only kills microbes that presents on the apparatus only and not on the materials. To make sure the plant and other materials is secured, other sterilization method has been conducted. For an example, nutrient media can be sterilized by two-way, which is autoclaving or membrane filtrations. But it is safer to use membrane filtration because it does not break down most compounds inside the media. Plant material can be sterilized via surface sterilizations using alcohol. It does kill microbes, but if the explant is juvenile or soft, a mild treatment is more preferred. Additional supplements also convenient to use in this purpose. Sodium hypochlorite, chlorine dioxide, and hydrogen peroxide are common use chemicals for this method, particularly compared to those compounds with similar properties of antimicrobial and antibiotics. Hydrogen peroxide is the most recommended chemicals as it has a minor side effect compared to sodium hypochlorite that may cause dehydration towards the explants, and chlorine dioxide, which is flammable and extremely powerful chemical. Overall, hydrogen peroxide is an excellent chemical for sterilizing explants, equipment, and surfaces in any lab, as it has few side effects compared to the other two. As a conclusion that can be drawn, sterilization is an important aspect to ensure the benefits of the plant tissue culture of Herbaceous plant can be optimized. However, this procedure still lacks in terms of their efficacy in maintaining the explant growth. Thus, sterilization procedure must be taken seriously, and further study is the only option to ensure the development of the tissue culture field.